You are welcome to my another session of cytology class. In this session, we are going to discuss about cell division with a reference to meiosis 1. The production of offsprings by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called as meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis involves two sequential cycles of nuclear division, that is called meiosis 1 and meiosis 2. But only a single cycle of DNA replication takes place. Meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase of cell cycle. Meiosis involves pairing of homologous chromosomes and recombination between them. Four haploid cells are produced at the end of meiosis II. The meiotic events can be grouped under two phases, that is meiosis I and meiosis II. The meiosis I consists of phases like prophase I, metaphase I, anaphase I, and telophase I. And meiosis II consists of prophase II, metaphase II, anaphase II, and telophase II. This is the schematic representation of meiosis. Different stages coming under meiosis I as well as meiosis II. The meiosis I, prophase I, metaphase I, anaphase I, and telophase I. And in prophase I, it consists of substages like leptotene, zygotene, pachytene, diplotin, and diakinesis. Whereas in meiosis II, it is only prophase II, metaphase II, anaphase II, and telophase II. The meiosis I, which is also called as heterolytic or reductional division. The first phase is the prophase I. Here, it occupies the longest duration in meiosis I, and it is divided into five substages or subphases like leptotene, zygotene, pachytene, diplotin, and lastly diakinesis. Leptotene. This phase starts immediately after interphase. The size of cell and nucleus increases. The chromosomes appear as long, uncoiled, thread-like in structure, bearing many bead-like structures and are called as chromomeres. The nuclear membrane and nucleolus remain as it is. Zygotene. This is the second phase, subphase in prophase I. Here, homologous chromosomes come closer and starts to pair up along their length. The pairing of homologous chromosomes is called synopsis, and the paired homologous chromosomes are referred as bivalents. The homologous chromosomes are held together by ribonuclear protein between them. Paquitin. In this subphase, the chromosome becomes shorter and thicker. Each chromosome of the bivalents splits longitudinally to form two chromatids, such that bivalence is composed of four strands and is known as a tetrad. The process of crossing over starts. The crossing over, that is a small fragment of chromosome exchange between the two non-sister chromatids of the bivalents by breakage and rejoining mechanism. The crossing over is the most important genetic phenomenon of meiosis, which causes a variation in genetic characters in offsprings. Diplotin. In this stage, crossing over takes place. The bivalents, or that is the chromatids, repel each other. Homologous chromosomes begins to separate, but separation is not complete. They remain attached to a point with a knot-like structure called chiasmata. The number of chiasmata varies. Depending upon the number of chiasmata, the chromosome appears different shape. One chiasmata cross-like. If there are two chiasmata, it looks like a ring. If more than two, it looks like a loop. The nuclear membrane and nucleolus begins to disappear at this subphase. Diakinesis. This is the last subphase in prophase 1 of meiosis 1. The chiasma moves towards the end of the chromosomes, that is the tetrads, due to contraction of chromosome, lastly slips over, separating the homologous chromosomes. This movement of the chiasmata towards the end of chromosome is called terminalization. By the end of the diakinesis, the nuclear membrane and nucleolus get completely disappeared, and the chromosomes are free in the cytoplasm. And lastly, spindle fibers begin to form. Metaphase 1. In this stage, the spindle fibers organized between the two poles and get attached to the centromere of the chromosomes. The chromosome moves to equator. The bivalent chromosomes are arranged in the equatorial plate in such a way that two metaphasic plates are formed. And this is anaphase 1. In anaphase 1, the spindle fibers contracts and pulls the whole chromosomes into the polar region. The separated chromosome is known as dyad. No splitting of chromosomes occurs so that centromere of each homologous chromosome does not divide. Thus, chromosome number of the daughter nuclei is reduced to half. Now, the separated chromosomes move towards the opposite poles. Telophase 1. Here, the two groups of chromosomes form at each pole and organize into a nuclei. The nuclear membrane and nucleolus reappears. The chromosomes get uncoiled into chromatin thread-like structures, and the spindle fibers disappear totally. Then in cytokinesis 1, it occurs by the cell plate formation, which divides the cell into two daughter cells which are haploid in nature.